Good morning, everyone. Welcome to this week's Department of Medicine Grand Rounds. Before we get started, a few housekeeping points. Please make sure your mics are muted during the presentation. Also, we kindly ask you to hold your questions and comments till the end, which time you may unmute yourself or put your questions in the chat box. Lastly, the CME credit on the screen will be displayed at the beginning and at the end of the presentation. For today's speaker's presentation, I'm going to pass it along to Dr. Sominathan.
Good morning, everyone. I'm really excited today to introduce a dear friend and colleague as our 2024 World AIDS Day speaker, Dr. Marina Caskey. Dr. Caskey has an amazing CV and resume that I can't cover in the brief time that we have. But to summarize, she finished her medical school at the Federal University at Sergipe in Brazil, and then did her residency at St. Luke's Roosevelt, and her fellowship training in ID at New York Presbyterian Weill Cornell, and currently serves as an ID physician at Weill Cornell, and she also is an elected member at the American Society of Clinical Investigation. Relevant to the talk today, she is a professor of clinical investigation at the Rockefeller University, where her research focuses on developing and clinically assessing novel and innovative immunotherapeutic approaches for combating infectious diseases, particularly HIV. She has led a multitude of early phase clinical trials to assess the safety and effectiveness, particularly of broadly neutralizing HIV antibodies, and currently serves as the chair of the HIV Reservoir and Viral Eradication Transformative Science Group at the ACTG, which is part of the NIH. And her work has really revitalized this entire area of HIV research, which had previously been sidelined because the first generation of antibodies really did not show a whole lot of promise. But currently, I feel like we are on the threshold of a whole new era in the future, where these bNAbs look to be one of the most promising and exciting strategies for achieving HIV remission and as potential alternatives to ART treatments for both HIV treatment and prevention. So with that, Dr. Caskey.
Hi, good morning. Thank you, Shoba. That was very kind. It's a pleasure to share with you our work on broadly neutralizing antibodies against HIV, which I'm going to refer to as bNAbs. So what I'm going to try to do here is to talk about so these are all still investigational molecules. So I'm going to discuss their potential roles for in the spectrum of prevention therapy and HIV cure. I'll share some of the results that we've had in our clinical trials, and then I'll finish with a focus on cure strategies. So as you all know, we've made huge strides in combating HIV infection. And this really began by the identification of HIV as the cause of AIDS, followed many years later by the development of triple therapy or antiretroviral therapy, where in the beginning, these regimens were very complex, but now we have a multitude of options of one pill once a day. And then really now we have entered the era of long-acting injectables, which really will change management of HIV in a big way. In terms of prevention, we know that you can prevent HIV transmission by using antiretrovirals, and that is called PrEP. And then very importantly, we also know that in people with HIV, when there is suppression of their viremia, there is also very low risk to no risk of transmission. So, and then more recently, and this is something that Shoba is very involved with, PrEP has really entered a new era where this new antiretroviral called lenacapavir, tested in now two purpose studies in different populations of people at risk to acquire HIV, showed like 100% efficacy in preventing infection. And then what's special about this drug is a new class of ARVs, and it has very long half-life. So this now gives us the opportunity to have PrEP that is administered only every 6 months. So the landscape of HIV prevention will be changing very drastically, we hope. However, despite all of these strides, globally, the incidence of HIV remains very high. Last year, there were more than 1 million new infections, and we are still, unfortunately, far from an effective vaccine. And then there's been, as I mentioned, these big strides in terms of development of PrEP, but the rollout of PrEP globally remains below what the targets are. In terms of therapy, people now live long, healthy lives with HIV, but that requires treatment that is lifelong. And HIV continues to be associated with comorbidities and ongoing stigma. In addition to that, ARVs are associated with very high costs. And in this graph here is showing its funding for ARVs globally. And then it is expected that we are going to have a huge gap in the next several years in terms of numbers of people that are on ARVs and then the available funds for ARVs. And unfortunately, this may change even further with the changes in our nationally that we are going to see starting next year. So in this context, what we have been trying to address is how can will antibodies have a role in HIV prevention therapy and then in the future HIV cure? So as you all know, the concept of passive immunization, which is the transfer of antibodies from a convalescent individual to prevent or treat infections, is something that has been around for a very long time. This started with transfer of diphtheria serum, and this actually led to early Nobel Prizes with this concept where you can prevent or treat infection with convalescent plasma. And this was revisited just a few years ago in the context of COVID. But beyond convalescent plasma, where you transfer a polyclonal serum, so where you have antibodies that are of different activity, another big development in the last decade or more is that the ability to develop monoclonal antibodies. So here you select antibodies that you know very well that are selected but based on their specificity and their activity, and then you generate these monoclonal antibodies to be given to people to either prevent or treat infection. And then a lot of what we learned in terms of HIV, the targets on the HIV envelope to antibodies, how to clone more effectively, etc. So all of these developments that had happened under the scope of HIV research really allowed the entire field to pivot to developing antibodies to SARS-CoV-2, now 5 years ago. And in return, what we have learned with the experience with SARS-CoV-2 is that these monoclonal antibodies were then administered to a very large and diverse population. So we learned a lot about the safety of antiviral monoclonal antibodies, and then we've learned, as expected, that they are very safe. And then COVID allowed us to see that they are in fact able to prevent and treat as long as that there is a good match between the antibody and the strains at the time. So in the context of HIV, how are these antibodies discovered? So the target for neutralization on HIV is the envelope trimer. So this is the HIV antigen that is on the surface of the virus. So that's available to be seen, to be detected by the antibodies. A few of the challenges with HIV envelope is that it's sparsely expressed on the surface. So the antibodies, the immune system doesn't have a lot of targets. In addition to that, these envelopes are hidden by a very complex shield of sugars, of glycans, so they are hard to be accessed, and they are very diverse, orders of magnitude more diverse than SARS-CoV-2. So despite all of these challenges, a subset of people with HIV, usually 2 to 3 years into their infection, their immune systems go on to develop these broadly neutralizing antibodies, which are antibodies that are able to neutralize, to block infection from different clades, different subtypes of HIV. So there are methods that were developed where you select people that have high neutralization titers, and then you sort their single B cells so that from those B cells, you can clone, you can generate monoclonal antibodies in the lab, create libraries of hundreds of these antibodies, and test very carefully against panels of viruses to then select the antibodies that have most activity and then greatest breadth. So through these types of methods, first we learned the different neutralization targets on the envelope, and they are highlighted here in different colors. So these are the areas of the envelope that are susceptible to neutralization. And then all of these numbers in different colors here, these are the BNAbs that now have entered clinical trials. So these are all the ones that are highlighted here, they fit this criteria of being broad and highly active. However, what this heat map here is trying to show is that even though they're very broad, there is still gaps of activity. So what this heat map shows is the neutralizing activity of different BNAbs here on this side on the left. These are the list of different antibodies, and then tested against virus strains from different clades. So here clade C that circulates in Africa, B that circulates in the United States, and an AE that circulates in Southeast Asia. And the point here is to show that even these new generation antibodies have these areas of gap here shown in white or green. So what this shows is that even though each individual antibody is broad, they will most likely, we know now that they have to be used in combination, just like with ARVs. So now that we have these tools and these clinical products that can be tested in clinical trials, the field as a whole has been trying to address these questions. Where can they fit in prevention? So we think that they can be alternatives to long-acting ARVs. And in contrast to long-acting PrEP, the cabotegravir, one of the advantages of BNAbs is that it acts on a different target on HIV, so it doesn't carry the risk of selecting resistance to the mainstream of treatment currently. In addition, because of their safety profile, they may be particularly helpful and interesting to use in the context of perinatal infection. And then I'll talk a little bit about this phase 2b study that was concluded a few years ago now with one of these antibodies. Again, for therapy, their advantage is their safety profile and the fact that they are long-acting, so dosing can be given every 6 months. And then cure, we will talk about that at the end, but it has to do with the fact that the antibodies have the Fc domain, the bottom part of the antibody, which allows these antibodies to tag infected cells and then connect and then alert or bring in other arms of the immune system. So the AIM study was this phase 2b study that happened. There were two parallel studies that tested one of these monoclonal antibodies called VRC01. One study happened in sub-Saharan Africa and enrolled women. And then the other study happened in the Americas and Europe and enrolled MSM and transgender women. In this study, the participants were randomized to receive placebo or receive different a low and a high dose of the antibodies. And the antibodies were given intravenously every 8 weeks for a total of 10 doses. So this was a success in itself to be able to roll out this very large study in different parts of the world. Unfortunately, the overall study did not show to prevent HIV, but in a subset analysis, what was learned, and it was very important, is that among the viruses that were very sensitive to the antibody here in this light blue, the efficacy of the antibody was of 75%. So what we learned from these studies is, number one, we could prevent HIV through monoclonal antibody as long as the antibody has adequate activity against circulating viruses. And this finding was regardless of gender or region-specific clades. So it could be active in different areas of the world, again, depending on the circulating strains. And then we also learned that the assays used to characterize the antibodies in the lab can also predict outcomes. But how should we look at the results of this study? Should we see it as a glass half full or half empty? So because at the end of the day, the study did not achieve efficacy at the entire study population. So now looking to the future, the question is, will we find the right combinations of antibodies to provide greater protection through this strategy? And what will be the roles of antibodies now that we are really entering the era of long-acting ARVs for preventions? And these are things that are questions that are currently unanswered. And you know, Shoba at the HPTN and HVTN, they are very busy trying to figure out how to continue to test these molecules for prevention. So now back to what we have been able to do here at Cornell in Rockefeller. We have worked with 2 of these antibodies where they're illustrated here in this structure, where you know, this beige and pink area illustrates the envelope, and then the red and the blue are the structures of the antibodies where they bind to envelope. So this is really just to illustrate that the 2 antibodies bind to different sites on the envelope, so they have complementary breadth of activity. And then we see this when this is tested in vitro. These are neutralization curves. So this is done in the lab where you give different concentrations of the antibodies and you see if you can block infection against different strains. The y-axis is the breadth of activity, and then the x-axis the potency. And then the bottom line is when you combine the 2 here in red, you achieve greater breadth and potency. But note that we are not achieving 100%. So even though these antibodies are very active, we still don't reach 100% coverage. So the studies that we've done, they have done here at Rockefeller, which is all the way in the east side of Manhattan, right next to Cornell, which is the building here. And then we have, it's a research institute. We have this small research hospital where we can only carry out protocols. And this is where we do our clinical operation. And then we work very closely with Michelle Nussenzweig's lab to study the samples from these study participants. So in our studies, we first wanted to ask the question, are these antibodies safe? What's their pharmacokinetics in humans? And then do they really have activity against circulating viruses in people with HIV? So the first studies, we tested either antibody alone or the 2 of them in combination. And the participants received a dose of the antibody at increasing levels, starting at a low dose up to a higher dose, which was our target dose. And then they're followed over time, where we monitor their viral loads, their CD4 counts. But importantly, we also looked at the virus and how it evolved over time under the pressure of the antibody. And what we found, these are all viral load curves where each line represents an individual, and this is their viral loads over time after receiving the antibody. So when we gave either antibody alone, we see that there is this decline in viremia in both cases. However, over time, the virus returns to baseline because there is selection of resistance, just like we would see with a single ARV. And these first generation unmodified antibodies had half-lives of about 2 to 3 weeks, which is what's expected from an IgG. When the 2 antibodies were combined, we are able to lower viremia further and then maintain the viral load below baseline for a longer period of time. However, even with these 2 antibodies, we're not able to achieve very complete full viral suppression, except in the individuals that had low-level viremia to start with. So what that says is that the 2 antibodies are not equivalent to the triple combination ARVs that we currently have. So in the context of viremia, it may be that the antibodies won't really have a role if they are given alone. But importantly, we do not see de novo resistance arise when the 2 antibodies were given. So going to this piece about selection of resistance. So in all of our studies, we had participants that responded to the antibodies and then participants who did not have a response. So even though they had not seen this particular BNAb in the past, because they had been living with HIV and their viruses had been co-evolving with their own antibodies, it's not unexpected that there would be a subset of individuals that would have viruses that would have escaped due to their own autologous antibody responses. So what we did is to see what type of resistance mutation or selection this particular antibody was given was to sequence envelopes at the beginning and then at 4 weeks and later after the antibody had been given. And what this is all illustrating is that the percent of the frequency of different envelope sequences over time, where gray represents the wild type, so the sequences that are known to be sensitive, that have the right amino acids in the target site of the antibody. So you see all these participants had sensitive wild type viruses to begin with, but after the antibody pressure, these different strains of envelope, they arise. And then there is a loss of activity to the antibody. But the other part that is interesting is over time, the wild type returns, which is an indirect way to suggest that these resistant viruses, even though they were selected for, they seem to be less fit than the wild type viruses. So that is not surprising, but it's also not great to see. So one obvious next question was, if we select resistance to this class of antibodies that binds to the V3 part of the envelope, what does that do to other classes of antibodies? So what this here is showing is in gray, again, these are viruses at baseline, and IC80, so the higher, in this case here, the higher the bar, the more sensitive to the antibody the viruses are. And then you see that against 1074, in an equivalent other antibody to the same epitope, there's resistance, there's selection of resistance strains after exposure to the antibody. However, when tested, the same sequences tested against antibodies that bind to other epitopes on envelope, you see that there is no selection of new resistance to these other antibodies, what tells us that the combination should be more effective in preventing resistance from arising. So we saw that in viremia, we learned that the antibodies are in fact active in vivo, but they were not sufficient to really cause full virus suppression. How about if we use the antibodies to replace conventional ART, where we would switch participants from their current ART regimens to receiving antibodies? So now we've done this study several times with some modifications. They all have the same principle. We enroll individuals that are doing well on ART. We give them doses of the two antibodies, and then we have now repeated these doses over a course of 6 months, and then they discontinue ART. So during this time here in blue, they are only under the exposure of the two antibodies without any ARV. And then again, we look at what's happening to their envelope sequences, in this case, in the provirus, because they are aviremic. So if we were to do the same experiment in someone who had been on ART and we just withdraw ART, the majority of the participants would have rebound viremia within a couple of weeks of being off ART. When we tested this with 3BNC alone, we were able to maintain suppression just for about 8 weeks, but then there was selection of resistance. However, when we gave the two antibodies here in this blue line, we were able to maintain viral suppression, in this case, viral loads less than 200 for a longer period of time, a median time of almost 4 months after the last dose of the antibody. So it showed that not everyone responded, but the ones that had sensitive viruses to begin with, we are able to successfully maintain suppression as long as we had enough antibodies on board. And then we have now expanded this to giving the antibodies over a period of 6 months in the context of participants who had started ARVs during chronic infection versus participants that started ART during early infection. And then we find similar results where about 75% of the participants, without having been screened for sensitivity of the antibodies, are able to maintain viral suppression as long as we keep the antibody levels at a therapeutic level. So this is all great, but it's inconvenient to give antibody infusions every month. So one other development that happened was to modify these monoclonal antibodies by 2 simple amino acid mutations in their Fc domain. So the introduction of these changes modifies how the antibodies are metabolized. It protects them from going into the lysosomes and being broken down. And then the result of this modification is that we are able to extend the half-life by about 3 or more fold. So in this graph here, we are showing the levels of antibody of the original parental non-modified 1074. You see after a dose within it falls below this threshold of 10 in just about 4 to 6 weeks. And this is in contrast to the modified version where the levels are maintained for a much longer period of time. So this has now allowed the ability to dose these antibodies very infrequently. And this, in fact, is now moved on to being for these long-acting antibodies are now being developed by Gilead, a pharmaceutical company that licensed these two antibodies, and now they're testing the two of them in combination with their capsid inhibitor. And then, in fact, their early studies suggest that this triple combination of one ARV in the two long-acting antibodies may be a viable option of therapy every 6 months. So from our early studies, we saw that the antibodies are generally safe. The parental original antibodies had half-lives of 2 to 3 weeks. They had in vivo activity and they lowered viremia. We were able to maintain virus suppression in individuals that had sensitive viruses. And now these long-acting antibodies allow infusions every 2 years. However, there is more that needs to be done. As I said again and again, the issue of pre-existing resistance remains. And this is being tackled by different groups with ongoing antibody discovery, engineering, where you have antibodies that have 2 or more specificities, selecting better combinations that could be fit for the different geographic areas, combining these antibodies with long-acting ARVs, and also working on screening assays that we could use to select the best participants or populations. So now switching to the last part, which is, besides working as ARVs, as long-acting ARVs, could these antibodies be able to have effects that ARVs do not have? Could they interfere with the latent viral reservoir or modify existing HIV immune responses with the idea that they could perhaps be part of what we call HIV cure strategies? So one of the main challenge of HIV, the reason that we need lifelong ARVs is that HIV remains in this pool of cells that harbor viruses that are able to replicate if ARV is removed, and that is called the reservoir. And these infected cells are very rare. They are not easily identified, so there's no surface marker that tells us that that's a latent cell, and they are maintained for life by different mechanisms. In addition to this virology problem of HIV, we also have an ongoing immune dysfunction that even though a lot of this can be improved by effective ARVs, it's not completely reversed. So there is still ongoing immune exhaustion in the context of ARTs, and then there's even when we do have immune responses, some of these cells are resistant to killing. So this is a daunting problem, but in the last several years, we have learned more about the reservoir that gives us hope and then perhaps gives us opportunities to intervene. So it used to be that the reservoir was thought to be very stable and untouchable, completely silent to the immune system. However, now we know that these clones of infected cells, they wax and wane over time. Some of them expand, some of them retract. And this is thought to be immunologically mediated because some of these cells are transcriptionally active, meaning that they are visible and can be targeted by the immune system. So with this idea that the reservoir can actually be targeted, there are different strategies that they're now being investigated. So the holy grail of HIV cure would be to achieve full eradication of HIV, where we would completely remove all of the replication competent viruses, either through gene editing, or you would bring them all out of latency, and then all of the cells harboring the virus would be eliminated. Or you could simply block and lock these viruses into the cell's DNA in a way that they could never resume viremia. But these are all, to achieve full eradication is a very lofty goal at the moment. So instead, the field has been focusing on what we call ART-free virologic control or remission. So the idea here is that we are not going to eliminate the reservoir entirely, but we would use different strategies to try to reduce this reservoir in a way, and at the same time, improve existing immune responses in a way that the immune system itself would be able to maintain viral suppression long-term without ARVs, the same way that we are able to do this against other chronic viruses. So how this would look over time. So, when someone acquires HIV, the viral loads increase very fast. With the elicitation of CD8 T cells, a viral set point is reached, but really it's ART that then shuts down ongoing viral replication, but it does not eliminate HIV. So the idea of these immunologic or curative interventions is that you could intervene during ART suppression in a way that you would try to modify the relationship of the virus with the immune system. And we think now that what would maintain virus suppression or low-level viremia in the absence of ART would really be a coordinated response that involves both the innate immune system and adaptive immune responses that would then shut down ongoing replication to lead to what we would like to achieve, which is we refer to as long-term control. So how do antibodies could play a role here? So antibodies can bind to free viruses, and then they are eliminated. So they can, in this sense, they can prevent new rounds of replication. But when we're talking about HIV cure, what we are interested in are in these other activities that antibodies have that are mediated to their FC domains. So they are able to target infected cells as long as they are expressing the antibody target, the HIV envelope on their surface, and these cells could then be killed. And they can also, by creating immune complexes, improve antigen presentation to dendritic cells and improve immune responses. And then there's experiments in preclinical, not in the clinical trials yet, that shows that antibodies can in fact clear infected cells. And studies in non-human primates also suggest that antibodies, when given alone during early infection or in combination with other immunologic strategies, lead to virologic control after the antibodies wash out. And this control is mediated by CD8 T cells, at least in part. But we don't still understand what differentiates the controllers from the non-controllers. And this is now what we are trying to address in clinical trials. So in the last 5-6 years, there's been interesting results from clinical trials where antibodies have been given with other drugs that are meant to reverse latency, to bring HIV out of the reservoir. And these strategies were given under ART suppression. But really what's been most intriguing and most interesting in terms of outcomes was when antibodies were given during in the absence of ART, during what we call analytical treatment interruption, or when they were given at a time the participants were initiating ARTs when there was decay in viremia. And we think that these results in ART initiation or during ATI, they have to do with the fact that there is more virus to antibody interaction that probably facilitates the antibodies from improving existing immune responses. Out of these studies, there have been some cases where the participants went on to become long-term controllers. And these are the viral load curves for these different antibodies, where, in black here are the viral loads. And then the colored lines are the antibody levels. In these 4 participants, even for a period of time after the antibodies were washed out, the viral loads were able to continue to be suppressed, and in some cases now for years. And this was also found in 2 other studies where the antibodies were given in combination with other immunologic strategies. So that's the clinical outcome, where we see that there is either a delay in viral rebound or a different viral rebound dynamics, and in these few cases where long-term control is achieved. But we don't really understand what's happening. And then one of the questions is this happening because the antibodies are interfering with the reservoir of infected cells or with host immune responses? And we don't have a lot of information on that yet. What we have seen in studies that we've done is when we look at the autologous, the person's own neutralizing antibody responses in a period of 6 months before and after they received the antibodies, we see that there was an increase in the breadth of antibody responses following exposure to a BNAb. So it seems that it accelerated the maturation of antibody responses. And in terms of T cells, we also saw that individuals that received the antibodies in the absence of ART, we saw this increase in HIV gag immune responses, both CD8 and CD4, over a period of time. It was not forever, but there was this increase in T cell responses. And lastly, we also saw that in individuals that received the antibodies over a period of 6 months, we saw that there is a decrease, even though it's a small decrease, in the number of cells with intact viruses. So these are all small, but indications that the antibodies are interacting with the virus and with the immune system in ways that ARVs do not. But however, these changes were not sufficient to prevent return of viremia, broadly speaking. We had these 1 or 2 cases, but we haven't been able to accomplish this more broadly. And this is a question that the whole field of HIV cure is trying to address. We are trying to really better understand the best timing for cure interventions during early infection, during chronic infection. We don't know how long these interventions need to be. And we are also trying to understand what's the best combination of interventions that we will need to use in order to achieve control. One interesting study was this study called eClear, done by investigators in Denmark. Here, they enrolled individuals that were ART naive, and they received either ART alone or the antibody 3BNC with or without a latency reversing agent. The treatment was, and this all happened at ART initiation. So the treatments here in this blue and green drawings, so it was a short-lived treatment period. The antibodies washed out, and the participants went into an ATI interruption period after a year of the intervention. And what they found was that, number one, in the participants that had sensitive viruses to the antibody to begin with, they had higher HIV-specific responses at the start of ATI. So immunologically, they had preserved anti-HIV T cell responses better than the ones that were either resistant to the antibody or did not receive the antibody. And this was correlated with the outcome that they saw after ART was discontinued. So here are again the viral load curves. Each line is one individual. The ones in red had resistant viruses to the antibody, so you did not really see an effect, whereas the ones in blue, they had delayed in return of viremia, and they had this blunted viral loads, including one individual that had full suppression and continues to be suppressed many years now after the intervention. So we've seen that we have effects on viremia. We also have some indication that both autologous antibody responses and T cell responses against HIV are modified with BNAbs, and also that the antibodies can impact the number of intact proviruses. But these are all small and perhaps short-lived effects so far. It looks like the interventions at ART initiation, as in the eClear study, seem to be particularly promising. So now in cure, there are many concepts that are being tested with these 2 antibodies, but also with other antibodies. And these are the type of strategies where the antibodies are being given during primary infection in combination with vaccines, with toll-like receptors, which are immune enhancing agents, and with different cytokines, again, to try to work together to enhance immune responses. And then this is a list of the different clinical trials that are happening. Again, testing the antibodies in the context of acute or early infection. or at ART restart, during suppressive ART, but with long exposure to the antibodies, or in combination with other molecules. And also now we are bringing these antibodies to Africa to try to address their activity against different viral clades. And then the last bit is we have been talking all about giving antibodies as passive administration, so where you give the antibodies themselves. But another area that has now some interest is, could we use vaccines that are being developed with the idea of eventually leading to the development of these BNAbs? Could they have a role in the context of HIV cure research? So the HIV field right now is very focused on these types of immunogens. They are called germline targeting immunogens. The idea is that they are designed specifically to expand B cells that initiate antibody responses that are on the path to become broadly neutralizing antibodies. And we have now clinical trials showing that this first step of inducing the first antibody response can be achieved. And now other studies are going to be tested if a combination with later immunogens could eventually lead to the development of BNAbs. And in the context of HIV, the idea is if we give a germline, start a new type of immune response in someone who is living with HIV, would the person's own envelopes, own viruses, serve as the boost to try to polish and expand those immune responses? Conversely, in someone who's living with HIV and already has this early type of antibody responses, could immunogens that can polish these responses lead to boost and expansion of these antibody responses to a point where you would have BNAbs generated through vaccination? And if we are able to accomplish this, it could be that these vaccine-induced BNAbs would be able to maintain virus suppression and also have those other FC effector functions. So one of these germline vaccines, it's called GT1.1, and this is a clinical trial that we participated in here in healthy volunteers, where they received the vaccine over a period of 6 months. And the key finding is that this vaccine could, in fact, expand the B cells that can then lead to CD4 binding site antibodies. And these antibodies that were generated to the vaccine, they bind to the HIV envelope, as in this structure here, from very similarly to VRC01, a broadly neutralizing antibody. So even though these antibodies are not as broad in potency as VRC01, they do interact with envelope in a similar fashion. So this is a result showing that this first step of vaccination can be achieved. And the idea is now, can a vaccine like this be now tested in the context of chronic HIV? So with that, I'll just finish. We've had multiple collaborators over the years. We do these studies here at Rockefeller, and then we work very closely with Michelle's lab for all of the experimental part. And then we have had collaborators through the ACTG, international collaborators in the various studies that we have done. And then none of this, I shouldn't forget, none of this could be done without the participants that really engage in these very complex studies, and they are very committed to being part of research. Thank you.
Dr. Caskey, thank you so much for this really tremendous presentation, and the science here is remarkable, and congratulations on all this great work to really advance treatment approaches for HIV. So if anybody has any questions, please feel free to speak up or raise the hand or put it in the chat. We're doing it from different locations this morning because of internet issues that we had, but I think we managed to get this done well. We had great participation from across our healthcare system. So thanks everybody for joining. I'm going to just start off and ask a question. The idea of targeting the reservoir is really fascinating to listen to some of that science. Is it that the reservoir cells are expressing some of the epitopes that the antibodies are targeting already, or is it really just the different mechanism of action there?
So the So the reservoir is not as, quiescent as we had thought. So so for example, if a clone of cells that have HIV is a clone that is specific for the flu, if someone is exposed to the flu, those cells are going to expand. And because HIV is integrated into the genome of those cells, it may be that from time to time, even on ART, that there's full expression, full generation of new virions, even if it's at low level. So that could make both as the virus is budding out of the cell, so then the envelope is now on the surface, could make them targets to the antibody, or if small amounts of virions are also being expressed, it could also trigger the immune system. But and this is happening all the time, but to low levels. So what we have seen is, so far, it seems that when you're on full virus suppression, we don't know that for sure, but I think that there is not enough of this activity happening to really engage the immune system with, or, through antibodies or through vaccination in a sufficient way. So that's why I think that doing these interventions, when the person is not on ARV, or when they are just starting ARVs, where the reservoir is still very dynamic, and there's still a lot of virus floating around, we may be more successful in that way because there will be more targets, which is exactly what you asked.
Thanks so much. Other comments, Dr. Swaminathan?
Thank you so much for this amazing talk. It's really exciting to be part of, cutting edge science. We enrolled our first 5386 participant a couple of weeks ago, and he's, getting his infusion that will be ready for ATI in a couple of weeks, which leads me to the first question, kind of more in the background necessarily, but it's one of the key steps in the HIV cure research, regardless of the modality, be it vaccine or broadly neutralizing antibody, is really, treatment interruption, that is in a clinical trial, not people stopping it. So do you want to comment a little bit on concerns that continue to have, despite all the data, physicians, providers, patients, community have a lot of concerns about that?
Yeah, so one of the challenges that we have in this, in HIV cure research is that, number one, we don't have, studying the reservoir is still very difficult because, as I said, these cells are very rare, and they are not easy to find because they don't have an obvious marker that we can look for. And that causes a problem in not understanding how effective your intervention was, because ideally, we would like to intervene and then have a marker that tells us, okay, you have had enough of an effect that now it's okay to discontinue ART and then watch for the clinical outcome, which is to see if viral loads were not going to return. But we don't have that. We don't really know what are the markers virologically or immunologically that would reassure us that someone could safely discontinue ART without having to experience viral rebound. So this leads to the fact that all of these studies, or the majority of these studies, have a component that include a period of time when someone comes off of ART. And then when someone comes off of ART, the vast majority of the participants will experience return of viremia. And with that, there is the concern of, risk of transmitting to a partner. And I think that as we have done more and more of this, now we feel more reassured that for the individual, him or herself, we monitor them very carefully during that period that they are off ART. We are monitoring their viral loads and their CD4 counts. So we know that we are not getting to a zone where we could trigger them to have opportunistic infections or anything like that. We also know that the vast majority of participants do not experience symptoms because of the return of viremia. So that's another reassurance. And then we also know that once they resume ART, the viral load suppresses very quickly. So within 4 weeks, all of them, the majority of them, have viral loads again back down to less than 200. So for the individual, I think it's a very difficult decision for anyone to make. When you're taking ART, you know that every day your viral load is undetectable, and you know that you are safe and then your partners are safe. Once you discontinue, it doesn't matter the intervention that we gave, we can never really know when the viral load is going to return. So it's a big decision to make. But I think the groups that are doing this, we are now becoming more confident to reassure the participant that viral load will suppress. But the part that we cannot fully address is this risk of potential transmission. And that's where counseling and having a very close relationship with the participants and their providers also come into play, because the person's behaviors and social interactions will have to change for that period of time. Lastly, the ideal thing that we would like to have as a companion to these clinical trials is to have at-home testing. Just like we had for COVID, when we all had COVID at home testing, we could go to parties, we could go to concerts, we could feel more comfortable and safe. Unfortunately, we don't have these tests for HIV viral loads yet. So that leads to participants having to return to clinic every week. So I think it's an evolving dialogue, both with participants, communities, and providers. But unfortunately, we don't have a way to avoid these ATIs if we want to understand the interventions and build upon these first results.
Thank you.
That was interesting. Now we have a question in the chat here from Dr. Vanga. Thank you for this great talk. Given the initial findings you presented, are you now including susceptibility testing as an inclusion criteria for future broad neutralizing antibody trials? And if so, has that impacted ability for participants in LMIC, given the cost and availability barriers of testing?
Right, so that's another big question. So the answer is some of the studies, most of the studies do screening. The problem is the current assay that exists that we can use in a clinical trial, that is a clear, clinical level assay, it's still unclear how predictive of outcome that assay is. Meaning that, sometimes the result looks like the virus is resistant and if the person is enrolled anyway, the person does remarkably well. And conversely, in some cases, it looks like the virus is sensitive and then the person rebounds quickly. And the reason for that is the reservoir, the diversity. We think about HIV very diverse out in the world, circulating strains, but the diversity even within a person living with HIV is extreme. So this assay, we send 10 mLs of blood. In that 10 mLs, you will have a few cells that will have HIV, and then they're able to sequence and test those viruses. But that often may not represent the diversity of viruses in a person. So that's why the test is not perfect, and we have these misses in both directions. So this is another area where there are many groups and investments being made in trying to improve the sensitivity testing so that the BNAbs could actually become a viable option. As far as LMIC, in the context of clinical trials, this doesn't become a barrier because this is built into the clinical trial. But going forward, yes, not going forward, thinking about antibodies, it's a whole other lecture probably of how would these expensive molecules become available in LMIC. There are, from an antibody manufacturing side of things, there are different technologies that being advanced where antibodies could be produced at a cheaper cost. But again, this is out into the future. And then the same thing for any therapy, even for the United States, if you needed to have a companion diagnostic, it makes it harder to use clinically for the cost and also for the practical aspects of it. So all of these things are kind of being worked on in parallel.
So interesting. Thank you. We have another question in the chat about reservoir cells. What about those other ones in the central nervous system? I guess, where do you look for them?
Right. So that's another Achilles heel of antibodies, where antibodies, regular looking antibodies, do not cross an intact blood-brain barrier well. So, and there are reservoirs in the CNS of HIV, and then there may be other sanctuary tissues where antibodies may not penetrate very well, although we do think that they do go into lymph nodes very well and into peripheral tissues. So in the CNS is challenging. There are strategies where they're being delivered in different ways, either through viral vectors, or they're, not the full antibody, but smaller versions of the antibodies. So there are people, or being delivered through cell therapy that could penetrate into the CNS. The caveat there is with anything CNS, you have reservoirs there. You would want, in this case, your antibody or antibody-like approach to get into the CNS. But how much inflammation, if there is going to be inflammation as a result, how much of that is going to be safe to happen, which goes into, what CAR T cells toxicities happen. So yes, these antibodies that I'm showing here, they do not have adequate levels of CNS penetration. There may be down the road other ways to do so, but it's going to have to be done carefully so that we understand if there are any associated toxicities.
Thank you. So a lot of challenges there. Certainly, I guess that's terminated. I just so you put a question in the chat here about have you observed, Dr. Caskey, development of antibodies against the neutralizing antibodies, thereby limiting their use, since they are being given repeatedly for effective treatment.
Right.
Yeah.
So we have in general these anti-antibody responses is something that can happen, and then in antibodies that are in clinical use, right, in rheumatology and cancer, this is one of the problems of some of the interventions. With these HIV antibodies, they are human antibodies. So the antibody. So for the ones that we are basically cloning from a B cell and then remaking it in the laboratory, they are fully human antibodies. So in these clinical trials, including in the AMP study that tested thousands of people, anti-antibody response was not an issue. There have been participants that have developed transient anti-antibody responses, but those were not functional, meaning they did not interfere with the activity or the pharmacokinetics or the safety, for that matter, of the antibodies. But we don't know, for example, with these long-acting antibodies where you have exposure over a long period of time and this slow decay, is that going to be more triggering of anti-antibody responses? And we don't know that. So far, it does not seem to be a problem, but everything is in the context of clinical trials where the exposure has been limited to 1 year, 2 years. So we don't know down the road if this is going to play a role. But there aren't any red flags for these passive transferred antibodies.
Thanks so much. Other comments, questions, Dr. Deaver?
Thank you so much, Dr. Caskey. That's just wonderful information. So I have a question. I'm very interested in the controllers, the long-term controllers in the study, and characterizing those, patients and their response. And how does that compare to the people that were controllers or patients that were controllers without getting, antibodies?
Yeah, that's a good question. So we don't know if they are necessary. So we don't know two things. We don't know if they were controllers by chance, and they would have been controllers on their own, or if the antibody really impacted their existing, virus and immune response in a way that helped them control, because the numbers are just so small that we can't really know for certain. So far, these are all anecdotes. They don't all look the same. So for example, for some of them, the virus in the reservoir is mostly in silent areas of the genome. So meaning that those viruses are probably less likely to reactivate, even though you can reactivate them in the lab if you stimulate those cells. But it seems that in some of these people, their own immune system had already selected out viruses that are mostly active. In others, they have very robust CD8 T cell responses that we think are playing a role. In others, their own autologous antibody responses are very active against the viruses in the reservoir, and probably if the virus is being able to replicate out of the cells, is not really blossoming into the blood because their own antibodies are clearing it. So they're all little, some of them have very small reservoirs, others have large, but of a very monogenic type of reservoir. So even though those viruses are there, probably means that the immune system is sufficient to keep it shut down, even though it's large. So we don't have a unifying theme, but I don't think there is any, but they don't, it's too early for us to know if the post-intervention controllers are going to really be immunologically or virologically different from the post-treatment controllers. We just don't have enough information yet.
Okay, thank you.
No, thank you. Thanks, Dr. Caskey. Dr. Tommy Nathan, any closing comments for us for World AIDS Day?
No, I think it's remarkable what we all have done in partnership with our community. And I think, and that's one thing that I think people living with HIV really were advocates for themselves and for their community, and they really pushed us as scientists and as clinicians to do more and more. And that is really why we are at this point where we have effective treatments and effective prevention. The challenge really is in us and in the healthcare system developing better implementation strategies to reach the populations that continue to be disproportionately impacted by HIV. And really, we have to do the work. It's not that, we have to get the word out to them and get the science to the community, because the science is there. We still have effect. We may not be at cure, but we are definitely have great modalities for both treatment and prevention. We just have to do a better job of getting it to the people who need it the most. Thank you
so much. I get goosebumps when I hear about this stuff, and having been an observer, of course, for a long time from the very beginning on this. And so, Dr. Caskey, thank you and congratulations on all the work and that you have done, and the science that you have brought to the table. That certainly, if not directly, absolutely a cure, we're getting pretty close to this. It's really just tremendous. And thank you for this really wonderful talk. Thank you, Dr. Swaminathan, for bringing this together for us, and for your comments about it, and how this is being translated out, directly to direct patient care. And it's really just wonderful to see it and hear about it. I want to thank everybody for joining this morning. Thank you, Dr. Deaver, also for helping putting this together as well. And really want to, again, Dr. Caskey, thank you for just a wonderful presentation, and wish everybody a great rest of the day.
Yeah, thank you all very much. It was a great conversation.